Okay, first page, sold. Okay, moving forward, these were the answers I came up with. If you got something different, that doesn't mean you're wrong, because the question says describe how the following are related to each other. Just because you may have come up with something different than what I came up with doesn't mean you're wrong. Put your hand in the air and say, here's what I thought. And we will discuss it. Okay, so first couple, here they are. Describe how the following are related to each other, DNA and RNA. If you went to the internet and asked Google, Gemini, ChatGPT, Grok, whatever the other AIs are, they may have given you a way more extensive answer than I have given you. But from my notes in this class, the only thing we've discussed that relates DNA and RNA to each other is the idea that they are both nucleic acids. That's it. The internet would have probably told you that RNA is formed from a copy of a section of DNA, which we will discuss when we talk about protein synthesis at some point in the next hour. But at this stage, for the homework I assigned, what I wrote down is really all you can say, unless you went to the internet. Adenine, thymine are complementary bases. That's how those are related. In a DNA sequence, they will pair with each other, is what is meant by the fact that they're complementary bases. How are complementary base pairings and hydrogen bonds related? We talked about this as well. Complementary bases connect to each other through hydrogen bonds or by hydrogen bonds. Nucleotides and nucleic acids. Nucleotides make up the structure of a nucleic acid. The next one is genes and alleles. How are those concepts related? Alleles are the specific representation of a gene. A gene might be, what does your hair do? This is the example I keep coming back to. And the individual alleles are, well, maybe the gene says straight. Maybe the gene says curly. Maybe the gene says wavy. Maybe the gene says uncontrollably frizzy. I don't know. But the genes are the specific representation of the overall general gene. Comments, questions, concerns? Evelyn. Phenomenal question. Matthew. Yes, that is correct. They're different versions of the same gene. Evelyn, I don't understand the question. How does a gene start? So eye color is complicated. Eye color is a combination of genes that interact in interesting ways. So at this stage, and with what we're going to talk about, I do not have a good answer for that. How specific traits are represented by combinations of genes is way beyond what we're going to talk about in this course. Like I said on the first day in this unit, we are going to oversimplify a bunch of stuff. And your specific question is more complicated. There are whole courses that answer your question. And then question F, we have not answered question F. There is nothing in the notes that I've given you up to today that is possible for you to answer F. So if you went to the internet, congratulations, you may have an answer to F. We are going to talk about F, hopefully at some point in the next hour. That's my goal. I didn't get to with C block, but I hope to with you guys, because they had those silly satisfaction surveys to do. The student satisfaction surveys that you guys have been being pulled out of class to do. Have you had to do them yet? No. Okay, so C block did theirs today. Okay, next set of questions. What's this? This is called a what? Karyotype. That is the answer to question 1. Why is this image prepared from cells that have stopped during metaphase of mitosis? Here's how I answered that question. You may have a similar answer. Because in metaphase, the chromosomes have been formed and lined up. The DNA has a longer answer. The DNA has been condensed into the individual chromosomes and have lined up across the center of the cell. They've let the cell do a lot of the work already, so they're organized. Comments, questions, concerns, does anyone say something different? As long as you said something similar to that, you're good. Comments, questions, concerns, going once, twice, thrice. Number 3. Explain how chromosomes are paired up and how the sizes are arranged. Here's how I answered that question. I could zoom this in, move it up for you a little bit, make it easier to see. I answered it by saying that they're sorted and paired by size, shape, and centromere location. That came from the place in our notes where we talked about homologous chromosomes and how you have pairs of chromosomes, how they have the same size, same shape, same centromere location, and those ones have the same genes. That's how they're sorted and paired into homologous chromosomes. Sorry, homologous chromosome pairs. That's where I pulled that information. The sex chromosomes is question 4. Where are they placed? You could answer this basically 3 different ways. I answered it with the word last. You could have also said at the end of the karyotype. Some people in block C said, can I say as the 23rd pair? Yeah. As long as you somehow signified that they are the last thing that gets placed in the karyotype, you're good. All sorts of ways to answer that. Question 5. What type of information can be gathered from this image about the autosomes? You'd have to remember what an autosome is. An autosome is which chromosomes? Where in the karyotype are the autosomes found? Does anyone remember? I hear flipping, which means nobody remembers. Yeah. Which means we probably didn't answer this question. The autosomes are the first 22 pairs. Basically, all of them that aren't sex chromosomes are called autosomes. So the only information that we're able to gather based on that karyotype is what I wrote down, that there are no problems with them regarding their number. For example, we talked about Down syndrome. We said, okay, well, Down syndrome occurs when there is a bonus gene. We can say that karyotype, that person does not have Down syndrome. There is no extra gene. There's no missing genes. There are 22 pairs of autosomes. Yes. You've got problems. I don't know what problems, depending on what gene is missing. But if you're missing a gene, that's a problem. That means you're missing 1/46 of all of your DNA, which is not a huge percentage, 1/46, because all your DNA is broken down into 46 chunks. That's why you have 46 chromosomes. So 1/46 times 3 billion. 1, 2, 3, 4, 5, 6, 7, 8, 9. So that is still, that number can't be right. 65,217,391 base pairs. That is not an insignificant chunk of your genetic code. If you're missing a gene, that's a problem. Sorry, if you're missing a whole chromosome, that's a problem. But, oh, you know what? I have a more complete answer to that question. But your chromosomes are in homologous pairs, which means you have copies. Like you've got this genetic code and this genetic code. Like 2 pieces of chromosomes have the same allele or the same sections on them code for the same gene section. So if one of them was just gone, you would only have the one there that would code for all of those traits. You'd still have codes for the same trait. You just wouldn't have anything to work with alongside of it. Does that make sense? You would still have a section that would code for certain traits. You just wouldn't have anything, it wouldn't have anything to work alongside. To compete with. So if mom has blonde and dad is redhead, and you were just missing the chromosome that you got from dad, you're going to be blonde. Because your mom's genetic code has nothing to compete with in you. Identify whether the person is, what shows that there's variation in chromosome structure? I mean, I just looked at the picture and all I came up with was there's different sizes and shapes. Like if all we're talking about is chromosome structure, it's not an overly deep question. You're just looking at the fact that how are they different? Well, there's different sizes, different shapes. That's all you can say at this stage. Question 7, identify whether the person is genetic male or genetic female. We talked about this. If a Y gene or chromosome is present, that person will code male. There will be other complications, certainly, but at least if they were genetically typed, they would type male, because that Y is present. There's a whole bunch of other problems with this person, because question 8, does this person have any chromosome abnormalities? The answer is yes. There are, if you looked, 2 X chromosomes. That's not supposed to happen. Massey, we good? Are we watching soccer again? Okay. The rest of the answers to your homework. How many sex chromosomes does this person have? They have 3. How many chromosomes do they then have? 47. They have 23 pairs and a bonus X chromosome. When do you think the abnormality in the chromosome number occurred? The only place you could get a problem with the sex chromosomes are in meiosis in mom or dad. So if the problem, because there's a second X, we don't know where that came from. Could have come from dad, could have come from mom. But in biological mother, biological father, there was a problem in meiosis. Because that's where the division here problem occurs. So either the egg in mom, while she was still an embryo herself before she was born, or in dad. And 12, why is the image helpful in making a medical diagnosis? Because as soon as we can identify that there's an extra chromosome, we know that there's going to be a genetic cause for at least some of the issues this person is experiencing. Daniel and Massey, what is going on? I have empty desks. If I need to move one or both of you, I can. There's 3 girls here who would love to shame one of you into behaving. That's what I would expect. Deep shame on them, whoever gets moved to you. Comments, questions, concerns? It's what I used to do with my grade 9s when they were messing around in class. I just go shove them next door to Mr. Keeley's class. And the grade 11s and 12s would shame them into behaving. Okay. No comments, no questions, no concerns. Cool, and flip the page. Let's talk about DNA replication. Here we go. Find me. Page looks like this. And we'll see how far we get. Do I have any coffee left? No, I do not. Okay, so last year, Science 9, whoever your teacher was, would have gone through the concepts of the cell cycle with you. They would have said interphase, cells live in the dream, cells, it's living, it's growing, it's copying. That's it. And then mitosis starts. Prophase, metaphase, anaphase, telophase, cytokinesis, boom, now you have 2 daughter cells. And somewhere in there, they would have brushed over the fact that DNA gets replicated. They would have said that, and they likely didn't go into any more detail than that. They just said, it happens. They might have even said a particular organelle is responsible for it. Does anyone know, did their science teacher in grade 9 tell them what organelle is responsible for the copying of the DNA? DNA is the string. It might be. I am not a biologist enough to know whether or not you're describing the thing I'm thinking of. The name of the organelle is related to the name of the thing that it's copying. DNA stands for deoxyribonucleic acid. So which organelle sounds like something in that word? The ribosome. The ribosome is the organelle responsible for copying your DNA. And now we're going to look at how that happens. You couldn't really talk about it last year because the concept of sugar phosphate group, nitrogenous base, the fact that there's complementary nitrogenous bases, as a general rule, that doesn't get discussed in grade 9. It's not part of the curriculum. Maybe your science teacher did bonus things, depending on who you had. I know Mr. O likes to go deeper into biology than other teachers do. Whatever, that's fine. But if you didn't, here it is. The replication is the process of creating an exact copy of a molecule of DNA. Now, your DNA is not one long chain inside your cell. Your DNA is broken up into smaller groups. How many of them? How many smaller chunks is your DNA broken up into? You know the number. Evelyn? No. No. No, it's a, when I say you know the number, it's not like you conceptually or you can count to this number. My daughter can count to this number. You like, it is related to another concept we've already talked about. Yes, Amitoj. 46. Why 46? Because there's 46 chromosomes. And when your DNA condenses, it condenses into a chromosome. It doesn't condense into one long chain. It condenses into a part of that chain. We call those parts chromosomes. So your DNA isn't one long chain. It's 46 smaller chains that condense into their own chromosome. So when your body copies, or every cell copies the DNA, it's not copying it in one long strand. It's copying it in 46 individual chunks, all at the same time, which makes it happen a whole lot faster. It's like if I asked you to copy down, I don't know, let's pick a large book, Les Mis, written by Victor Hugo. If you've never heard of the book, it's a fantastic book about the French Revolution. Really interesting. They made a movie starring Liam Neeson and Anne Hathaway and Hugh Jackman and Russell Crowe, and great movie. Fantastic story. The unabridged version is like 5,000 pages or something ridiculous like that. It's like I'm asking, if I asked you to copy the book Les Mis versus I get 5,000 people to copy out one page of Les Mis and you all copy it at the same time. It goes a lot faster when you copy it. Each of you get a chunk all at once. Same idea. So DNA gets copied. And as it's getting copied, it copies the whole genome, G-E-N-O-M-E. Your whole genetic code gets copied all at once in DNA replication. We're going to talk about a secondary process that does not do this. But in replication, the genome gets copied all at once and only once in the cell cycle. A human cell can copy all of its DNA in a few hours, which is pretty remarkable. With an error rate, this blank here is for errors, or error, of only one error, one mistake per 1 billion nucleotide pairs. That's pretty impressive. There are roughly 3 billion pairs of nucleotides in one complete DNA chunk, molecule, the whole thing. That's 3 mistakes total in the whole chain. That's incredible. Better than I could do, certainly. I don't know if you ever tried to copy down 3 billion things. I'm willing to bet you've made mistakes. And now each new molecule of DNA serves as a template, and I'll explain what I mean here in a second with a demonstration, as a template for the formation of its complementary strand. Each new DNA molecule, or each new molecule of DNA, contains one strand of the original complementary DNA molecule and one new daughter strand. And I'm going to demonstrate right now what I mean by this. So, DNA replication. It's hard to visualize, so let's visualize it practically. I don't know who this is. Actually, I'm pretty sure I do know who it is. Oh, he put his name on it. This is the attempt at origami DNA from a young man in the other class, who then crumpled it up and threw it at his buddy because he was doing a bad job. Luca. This is Luca's DNA attempt. Didn't work out so well. But he's created us an opportunity for me to demonstrate DNA replication. So thank you, Luca. So here's what happens. Here's your chunk of DNA. Inside your cell, let's pretend this is one whole molecule of DNA. Your ribosomes come along and they say, okay, cool, I need to copy this. So they don't grab scissors, they just kind of yank. And those hydrogen bonds give up. And what was the original DNA strand is now 2 halves. And now each one of these, this was good, workable, usable DNA. We know this because the cell didn't die at any point, right? The DNA is what codes your cells to do their thing, and because the cell made it to the end of its life functioning with this DNA, there were no mistakes. Great. So then the ribosomes are like, okay, cool. We need to copy this thing. Yank. Pulls apart. Because there are hydrogen bonds in between, they can pull apart without breaking the structure. The backbone is still intact. The individual nitrogen bases on each side are still intact. They're just not, attached to each other anymore. And then they go. They go, okay, you, ribosome buddies, copy. You, ribosome buddies, copy. And so now these sets of ribosomes are now responsible for collecting from somewhere in the cell, making the phosphate sugar backbone and corresponding nitrogen base. So start at the top of your chain. Tell me what you need. I am the cell. And we're making this thing. We're copying it. Go. What do you need? What's the first thing you need, Brooklyn? You need a C. Okay, cool. Click, and it clicks up. That's what I like this guy down here. How do you know you needed a C? Because it's the opposite of G. Cool. So then over here, we're going to click in the C. And it clicks. Okay, cool. What's the next thing you need? A T. Here you go. It clicks in underneath. What's the next thing you need? A C. Okay, Matthew, you do the same thing that I'm doing for these girls for Mullen. The pieces you need are over here. Tell them what you need. Send them over here and get some. Brooklyn, what do you need next? You need another G. Come on, get up. Walk. Matthew. Or one of you. Okay, keep going. What do you need? Okay, cool. You guys, Brooklyn and I are going to win. Okay, what do you need? You sure? Oh, we make our mistake. That one? What was it? C. Oh, C. We made our 1 in 3 billion mistake. You needed a T. Here's the T. Okay, what's next? Now you need the C. See, we demonstrated the 1 billion mistake. What's next? An A. Okay, cool. Here's your A. Whatever you need to know. Tell me what's next. Another C. Okay. What's next? You need an A. Where's an A? What color is A? A's green. Okay, cool. No.
There's no more.
There's no more A's? There has to be more A's.
Oh,
I need to see. What? Oh, you're holding A's in your hand.
I know.
The green one.
G.
Ah, you got an A.
G.
Cool. Okay, so now, for those of you who haven't, descended into coloring out, you've been kind of watching what's been going on here. It's kind of a race, but this is what happens at the end of interphase. The DNA gets ripped apart, and now you've got one team that's trying to put these things together, and another team that's trying to put these things together. And there's mistakes because there shouldn't be anything left over here. That's okay. Oh, you know why there's things left over here is because they were doubled up. They didn't make them full. So, but then we look over here. What do we have? Here, we've got a full copy of the DNA molecule we started with. Over here, we've got another full copy of the DNA molecule we started with. Half of each is what I provided at the beginning when I ripped the two chunks in half. And the other half was provided over the course of the building process. One piece at a time, click all the way down. Like you're going to a Lego box for the next piece, kind of one at a time. And now around each one of these, well, these would then get lined up in the middle of the cell. Your sister chromatids, half gets pulled one way, half gets pulled the other way, a nucleus forms around here, a nucleus forms around here, and now the rest of the organelles move over and they split cytokinesis and cell, daughter cell, or daughter cell and daughter cell. And we've copied the DNA successfully, starting with one strand of good DNA on each. Does that help with the picture at all? Just kind of watching it sort of happen. For those of you who are able to see, kind of watching it happen. You guys can bring your chunk and put it over here. Okay. So here's a picture version of the same kind of thing, so that you've at least got a copy of this idea in your notes. So let's start here. I'm going to grab a color, not just your pencil color, but find a color that works for this. The original DNA, I'm going to trace out here in green. Just kind of a rough outline of this is the path the original DNA took. And then here's the other side of that original DNA. And I'm going to make it obvious. If you need a color, come get one from me. Borrow one from a friend. Highlighter would work well here. Do not grab a whiteboard pen, please, because using them on paper will wreck them. And you've got the original is getting kind of unzipped here as it gets copied. And so down here at the bottom, I'm going to represent a chunk of each of these with these lines here. So you've got this side over here, and you've got this upper part I'm going to put over here on the right. And if we start down here at the bottom, we can start to see on the original, the pattern goes G. I'm going to start on the bottom over here. Oh, come on. Let's zoom out just a smidge. On the bottom one, it goes G, and then T, and then A, and then G, and then A, and then A. Oh no, hold on. G, T, A, G, T, G, and then T, and then another T, and then an A, and then a G. I'm only going to do the first 1, 2, 3, 4, 5, 6, 7, 8. I made it 1, 2, 3, 4, 5, 6, 7, 8. This is as far as I've done, for example. But then on the other side, we can look over here and say, okay, well, what have we got? We've got a C on the original. C, A, T, C, A, T, C. So these are two sides of the original. I made it this far here. And we can see that in the original, it goes together in the complementary base pairs. G with C, T with A with T, G with C, T with T with A with T, G with C. But then in the copying process, we can take a look at what's happening here. Ribosomes come along and then they click in the corresponding pieces that need to be added here. So what's going to go with G? Well, C is going to go with G, and A is going to go with T, T is going to go with A, C is going to go with G, A is going to go with T, A is going to go with T, T is going to go with A, and C is going to go with G. And each one of those pieces comes with a corresponding backbone piece. And now on this side, you've got a complete DNA. Well, over here, let's take a look at what's happening here. The ribosomes are going to bring in the corresponding pieces. What goes with C? G. What goes with A? Well, I need a T. What goes with T? I need an A. Then G, T, T, A, G. And all the way down. We're going to get that sugar phosphate backbone. Only one? Oh, it's a 2 chocolate egg day. Yeesh. And now look at what we've got here. G with C, A with T, T with A, T with A, G with C, A with T, T with A, and G with C. We've got 2 identical copies of the same DNA chunk. Comments, questions, concerns? And this is DNA replication. Clear as mud? Or are we following, picking up what I'm putting down? Someone say things are either good or bad. We're good? Okay, cool. Then what I'd like you to do is flip the page. And we're not going to do it right now, but I'm going to tell you, this page here is part of your homework. Actually, maybe I should give you a couple minutes to do this. Yes. Okay, 5 minutes on your mark, set, go. This page here it's exactly what we just talked about, but now you have to do it yourself. I'll give you a couple minutes, discuss it with your people. Crank out for me the DNA replication page on your mark, set, go. Call me over if you've got questions or need help. What's going on here? Okay, call me over if you. I'm surprised. There's one thing right at the beginning here. I'm surprised no one's got a question about. There's things up at the top that you need to fill in. Fill in the identification key. You need to fill in some letters up here. Yes, you can go to the washroom. What is what? What is there? It is. What does the D here stand for? Because what should this be? The backbone. So the backbone is made up of what? And what's the name of the sugar? That is a name of a sugar. What is the name of the sugar in DNA? What is the name of the sugar in DNA? It's the D in DNA. Deoxyribose. That's why this is given the letter D. Because P stands for phosphate group, D stands for not just the generic sugar, but the specific sugar molecule, deoxyribose. That's what that D means. How are you doing? Come here, how are you? Good. Are you just today? Or you got the vibe for how things go with me? Yeah. Oh, no, you can't point from your own voice out. That's
He's not here yet.
Careful. These are actually little gaps. There's nothing. There's the open air. There's nothing that connects these together.
For what? For C? Timeline. Yeah. So literally the first, like very first color. So literally up there.
put the pieces together. Unpause. Okay, let's look at protein synthesis. So DNA replication takes your entire genetic code, copies it once, then mitosis happens. No, there's nothing to write here. Don't write anything down in a heap yet. I'm summarizing what we did on the previous page. And now we've got protein synthesis. Protein synthesis, similar to DNA replication, but it is not the same thing. DNA replication happens once. Protein synthesis happens all the time. You need tons of different proteins to regulate a bunch of the different things that happen in you. Some of the parts of you we've already talked about are just pure protein. So that section of, or that protein synthesis is happening all the time. So this is different than DNA replication. Similar mechanisms, but fundamentally a different process. Here is, let's find a different color. Let's go purple, I guess. Let's use purple. After DNA replicates, mitosis is done. Now, interphase begins again in the daughter cells. After DNA replicates, the code is copied again. But not all of it, just pieces of it. Now your DNA starts to get used. It gets copied into, I'll say it gets, I'll make an ad here, copied in chunks. If you can fit that in. Into not more DNA, instead it gets copied, the chunks get copied into something called RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. I'll just put it underneath here. Ribonucleic acid. Zoom in here a little bit, make your life a little bit easier. And ribo, this piece right here, that is the fundamental difference between DNA and RNA. DNA uses deoxyribose as the sugar backbone. RNA uses ribose as the sugar background. There's no deoxy. So this is ribose as the sugar backbone. And it is not meant to be permanent. RNA is not a permanent structure. DNA is permanent, it's meant to be there forever. Ribonucleic acid is not. So DNA, or the RNA gets copied. DNA gets copied in chunks, makes RNA. And then the RNA gets translated into a protein. So after DNA replicates, the genetic code is copied in chunks into RNA and translated into a protein. Every one of those RNA chunks is going to code into potentially a different protein. So here's a picture of it. DNA, once it gets copied, now we need to start accessing the bits and pieces. So here's how I will demonstrate this. To make, I'll use this long thing here. To start proteins, and I'll put it on this middle table here. You know what? No, I'm going to go over here so that Johnny can see me. My back's not one student. Okay, so we've got our whole DNA chain over here. Table of Brooklands.
whole DNA chain. It's been replicated. This is one of the daughter chains. Now we need to use this thing that makes for making proteins. Now, your body needs a particular protein. Where in this chain is the protein? I don't know. Your body knows. It knows what chunks code for what, and so we need a particular chain, a particular piece of this chain. Maybe we need this guy right here. So at whatever location you need the DNA, that was where it unzips. So let's unzip it. These scissors aren't great. But let's say we need a chunk, and we need to unzip it going this way. And say, okay, we're not unzipping the whole thing, we're just unzipping this. We're just unzipping this piece. And while this is open and unzipped, we're going to start finding the chunk that we need, and then we're going to copy just that chunk. So I've got an AGC, AGTGTG chunk. So along comes your ribosomes. It's like, okay, we're going to code for this specific protein, not the entire DNA chunk. So I'm going to bring in the corresponding nitrogenous bases for this chunk of DNA. I'm going to take that RNA away, and I'm going to allow this DNA to zip back up, and then the DNA remains intact. I've just taken out a piece, I've translated a chunk of this into something I can walk away with. This chunk, copy it, I walk away. Now this is my RNA. Now I take my RNA and I say, okay, boys, make me my protein. And you go the same thing that you did before to make the entire DNA code, you would do, but just on a smaller scale, just for this protein piece. I'm not going to make you do that one already. But the idea is the same. Instead of making the whole chain now, you're just making the protein from this RNA chunk. Now, there's a second fundamental difference between DNA and RNA. I'm going to give it to you here. In DNA, let's make a list of the nitrogenous bases that exist in DNA. What are they? Adenine, thymine, guanine, and cytosine, or cytosine. Okay, these are the nitrogenous bases that exist in DNA. RNA has one fundamental difference. So let's make a list for the RNA over here. In RNA, adenine stays. Guanine stays. Cytosine stays. But there is no thymine in RNA. The thymine gets replaced. Why? I don't know. But the thymine does not exist in the RNA. The thymine gets replaced with a nitrogenous base called uracil. This guy here, for whatever reason, Evelyn, don't ask me why, I don't know the answer. We could Google it, I'm sure. RNA does not include thymine, it includes uracil instead. Sorry? The same places all the other nitrogenous bases come from. As your body needs them, it builds them. Like when you, so like in the DNA replication, you don't have just spare, your body's not just full of spare parts. As needed, they get constructed based out of the nutrients that you've consumed. So when RNA is being made to go ahead and code for a protein, uracil gets placed in to the RNA code, and that gets taken away. It's another thing that differentiates RNA from DNA. DNA has the deoxyribose sugar, RNA has ribose sugar, and rather than thymine, there's uracil. So these steps are also given specific names. You've got DNA, and the DNA might be the entirety of your code.
The DNA, a chunk of it gets copied into a single strand of DNA. So maybe your DNA represents a whole book. It's the book that makes all of you. But I don't need the book that makes all of you every time I need to make more hair of you. Maybe I'm your ribosome, and I'm like, well, Evelyn needs more hair, so let's start making the protein to make Evelyn's hair. I don't need to copy all of the DNA, I just need to copy a specific piece. So what I'm going to do is I'm going to transcribe. The process is called transcription. I'm going to copy a chapter. But for whatever reason, I don't copy the chapter down fully. I copy a chapter of notes. I don't copy down fully. I make little not tweaks, but I make notations of the chapter. So I take the whole book, I transcribe the chapter, and then I go out to my computer and I'm like, okay, cool. Now I'm going to take these notes and I'm going to rewrite the entirety of the page or the chapter using the notes I made to help me remember what I was doing. Does that make sense? I'm not allowed to take the, or it's like going to the library. You take the book off the shelf, but you're not allowed to borrow the library book because you bought the card. So you go and you're like, okay, I'm going to make a bunch of notes to help me remember what's going on in this chapter, and then I'm going to go home and I'm going to type it out on my computer. That process is called translation. You take your copy of notes and you rewrite the chapter word for word. And then you put that chapter to work. Maybe you're making more hair. Maybe you're making fingernails. Maybe you need more hemoglobin. Whatever the protein is, it's now doing work. This is protein synthesis. There's an extra couple steps in here. It's substantially more complicated than just DNA replication. If you've got questions while we're going, please ask. I'd like to make sure things are clear. I'm happy to clarify. Okay, so here we go. Let's fill in these blanks to talk this through. When needed, a cell accesses the genetic information coded within a single gene, the chapter I was referring to create proteins for cellular activity, whatever that activity needs to be. An enzyme called RNA polymerase. Polymerase is used to copy the sequence of the DNA's nitrogenous bases to create a single strand of RNA nucleotides. So the RNA polymerase is used at this step in transcription to make the RNA chunk of nucleotides that are needed to code for a particular protein. Next, this single strand of RNA is used to produce the correct sequence of amino acids to build the protein. Thus, the sequence of bases in DNA's genetic code will determine the sequence of bases on a strand of RNA, which will in turn determine the sequence of amino acids needed to make a protein. Now, if you've got friends in Mr. O or Mrs. Broad's science classes, well, I know Mrs. Broad does this. Mr. O started this, and then Ms. Broad took some of his stuff, and they actually go into a lot more detail of how this happens. I'm not planning on doing that because I want to still get to astronomy and physics. So I'm not going to take the time to go into it in more detail. That exists. You can, but there's whole biology courses on that. There's a section on astronomy in the Science 10 curriculum that I would like to teach. Yes. Yes. So, yes. So a condensed version of it is when DNA gets transcribed. It gets transcribed in chunks of 3 nitrogenous base pairs. So they'll take a 3-letter chunk. Get down here with me here. I'll zoom in on this so you can see what I'm doing. They'll take a 3-letter chunk, CTG, and they'll take another 3-letter chunk, ACG, and they'll take another 3-letter chunk, GAT. They'll take another 3-letter chunk, CAG, another 3-letter chunk, another 3-letter chunk, another 3-letter chunk, another 3-letter chunk, that one, that one, that one, and then there's the last one. So here there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Oops, sorry. Let's move this over a little bit. There are 12 3-letter chunks. Each one of these 3-letter chunks codes for a different kind of amino acid. RNA transcribes. Where everywhere there should have been a thymine, uracil goes in instead. So you can see here, it goes CTG, ACG. In the RNA, it goes GAC, that's good. But in the RNA, where there should have been a T that goes up with this A right here, that's where uracil gets plugged in. So A goes with U, C goes with G, G goes with C, G goes with C, A goes with U again. And then T goes with A, C goes with G. Anywhere where there was a T, you get this U in the RNA, and every single one of these codes for another amino acid, and then these amino acids get slapped together and you make your full protein chain. Comments, questions, concerns so far? I'm sure there will be when we actually stop to play with this stuff. So flip the page. There's another example right here. It's the same idea. You get these 3-letter chunks that start, so there's however many of them there are. You get this section of DNA that unzips. And so for this guy here, let's make the corresponding RNA chain. The RNA that's going to copy from this DNA is going to look like what? The very first thing is where we're going to get the RNA difference. In DNA, A would go with T. But T does not exist in an RNA molecule. So the very first letter down here in the RNA molecule that's going to code from this is going to be what? U. So the RNA is going to start with UAC. Is going to be the first thing that codes from these 3 letters. ATG is going to become UAC. A is going to go with U again. C is going to go with G. G is going to go with C. So it's going to start UAC, and then it's going to go UGC. There are no Q's in DNA, Mahip. If it looks like a G or a Q, it's a G. So it goes UAC, UGC. What's the next 3 going to give you? GAT is going to become what in the RNA? CUA. Very good. And then CAG is going to become GUC. Very good. In wherever you see it, because adenine is allowed to exist within RNA, a T in the DNA is going to give you A in the RNA. But T is not allowed to exist in the RNA, so the A in the DNA is going to give you the U in the RNA. A pairs with U, but T still pairs with A. Anywhere where there should be a T, you're going to get a U instead. But all the other letters are still the same. We're matching it with the top, yes. A with U, T with A. Yes. So I've got 1, 2, 3, 4 chunks done. That's these first 4. 1, 2, 3, 4. What I'd like you to do how do I want to play this out? How much time do we got left? 7 minutes. What I'd like you to do is I want you to finish off the RNA code, and I would like you guys to answer these 2 questions, and that is all you have to do for me for the rest of the block. There's no other homework beyond this. Finish off that RNA code and answer those 2 questions. And then you can pack up and be done for the day. So now we've talked about DNA replication and protein synthesis. On your mark, it's a giver. When the bell goes, you may go.